analysis. So that's going to be really the large focus of the talk today. I'd like to share a variety of different tools that we've developed over the years, as well as some application data that has some focus in immuno-oncology that I hope you'll find interesting and useful. So I'll start with one of my favorite quotes from the old days in molecular probes. Richard Haugland and his wife Rosaria co-founded molecular probes back in the 70s in their kitchen in Minnesota. The rest is history, and as an organic chemist, this was one of my favorite quotes from Richard about how one chemist can keep 10 biologists very busy. And I like this because it really underscores really where molecular probes came from. That is this idea of engineering fluorescence for cell biology. This notion that we can use chemistry to modify a variety of different fluorescent backbones to functionalize probes for a seemingly infinite number of applications. So if you're familiar at all with the legacy molecular probes set of assays and kits and technologies, you're probably familiar with the handbook on fluorescence labeling and detection. This is really an incredible source of information. I use it all the time to remind myself about how various fluorescent molecules work and their applications, and some great references to the literature. It can really be daunting, though, knowing where to start in designing an experiment. So I tend to break down these approaches in fluorescence labeling detection into three major categories. I find it useful just to think about designing experiments. Now, it's not to suggest that any one of these approaches is better than the other. It's really just to suggest that there are advantages and caveats to each category that are useful to be aware of when you are designing experiments in fluorescence. The first major category is really that classic molecular probes approach, the fluorescent dyes. These organic molecules are wide in variety. They range across the fluorescent spectrum from the UV to the infrared. Many applications in both living and fixed cells. And traditionally, one issue with this approach can be photostability, right? Especially in fluorescence microscopy, where we tend to expose our samples to fairly intense levels of light. We've innovated, again, through chemistry over the years to develop probes or fluorescent dyes like Alexafluor or Alexafluor Plus fluorophores that are particularly photostable and bright. To mitigate this, we've also taken approaches in protecting samples that are fluorescently labeled through the development of various anti-fade mountants. Many applications in this category, of course, we're all familiar in immunolabeling with the use of fluorescent conjugates of antibodies or even streptavidins. Some basic applications in cell tracking, that is really just to label cells, whole cells, for a variety of different applications, be it angiogenesis, wound healing, just really knowing where a cell is or populations of cells are and where they might be going or how they might be behaving. We can also functionalize fluorescent dyes so that they're responsive to a variety of different intracellular phenomena. We can design them to be responsive to changes in pH or any other number of ions, to detect changes in voltage across intracellular membranes. We can even design them so that they selectively accumulate in intracellular compartments. So we have a wide variety of organelle probes that we've developed. In the area of high content screening, it's also important to appreciate the value of being able to robustly label our cells, be it through nuclear stains, plasma membrane stains, or whole cell stains. It's really important to robustly label cells so that they can be identified as objects using automated software to identify them as such, and then allow us to analyze our images for regions of interest within those objects. So the second category of fluorescent proteins, of course, exquisite tools for interrogating a variety of different aspects of cell biology. A little bit more limited on the spectral range. These, of course, are tools that we express in living cells, but many of the fluorescent proteins are adequately fixable for endpoint analyses. A variety of photostabilities in the different fluorescent proteins that are available to us But generally speaking, these tools have a lot of value in enabling longer-term live cell imaging. So we can add valuable temporal resolution into our imaging experiments. I'll speak to during this webinar in terms of what I'll phrase as functional biosensors, that is, expression of fluorescent proteins that have been modified in different ways to be responsive to changes in the intracellular environment, or expression of fluorescent proteins as fusions to localization sequences that allow us to target these fluorescent proteins to specific compartments or structures in cells. The last category are the fluorescent quantum dots, really interesting materials that are comprised of a core and a shell. As the schematic indicates, the larger the core, the longer the wavelength emission. These are useful tools in many applications, both living and fixed, not unlike organic dyes. But what makes these materials very interesting and useful is their incredible brightness and photostability. Again, many applications like fluorescent dyes, including conjugates of antibodies, cell tracking applications, as you can imagine, with brightness and photostability being their most redeeming attributes. These are useful in longer-term cell tracking applications. They've also found a niche in vivo imaging, such as tracking vasculature or even tumors through skin of whole animals and whole animal imaging experiments. And they are sufficiently electron dense to be useful in EM and CLEM applications. So having set the stage for these different approaches we can take with fluorescence labeling and detection, I'll now switch gears and present a number of different assays and probes that we've developed, but I'll do so in context of what I'll refer to as the cell health continuum. So this is important to all of us as cell biologists because we know that changes in cell health can really be indicative of useful information. Changes can underlie progression to a disease, of course. Understanding various parameters of cell health in a population can tell us something about the effectiveness of a potential drug or perhaps the function of a gene in knockout studies. And it's a continuum, in my view, because it's important to consider more than just whether or not a cell is live or dead. There's a lot going on in this continuum. Maybe cells have been stressed and protective mechanisms are triggered, or perhaps once a commitment to cell death is made, then perhaps mechanistically it's important to understand how cells have died and what's going on after they've died and what is what's involved in terms of a cleanup phase. So when we consider cell health as more of a continuum like that, we can start to consider various aspects of cell health and cell stress and cell death that might help us understand more in interrogating populations of cells. So just in terms of simple viability, there's a lot to consider, right? Whether or not mitochondria are polarized or the cytoplasm is still reduced, are QC mechanisms for RNA and protein still intact? Are cells proliferating as they should in a healthy population? And even morphology can tell us a lot about the health of cells in populations. So if cells become stressed, what are these, not necessarily a commitment to death, but what are these pre-lethal indications of cell stress? Is there loss of those QC mechanisms? Have mitochondria just transiently depolarized? Is there oxidative or nitrative stress or even dysregulation of cell cycle? And if stress has happened, then we might be interested in understanding what are some of the mechanisms that cells will trigger under cell stress, perhaps autophagy or DNA repair mechanisms. If cell death is happening, again, it might be interesting to know whether or not apoptosis or necrosis is happening. And there, of course, are various aspects of these death pathways that we can interrogate to understand more. Of course, there's always the crude measurement of cell death that's very useful, just simply loss of plasma membrane integrity. And of course, we've developed a number of probes over the years to be able to do that. After cells have died, what are the immunological consequences of cell death, right? It's important to be able to, in a regulated fashion, clean up cells that have died by programmed cell death. Otherwise, what are the inflammatory responses that we can understand in a population of cells as they die? Okay, so let's focus in on a number of these aspects as I walk you through assays and probes that we've developed. Let's start with morphology. So morphology, like with the general approaches to fluorescence labeling and detection, I think about them in three general approaches. The first is a fairly obvious approach. We can exploit the often very good specificity of antibodies, in this case against resident proteins of various organelles or cell structures, to visualize a variety of different structures in typically fixed cells. This is achieved typically through the use of primary antibodies, then subsequently detected with secondary antibodies conjugated to a fluorophore. But there's also a variety of different primary antibodies that are directly conjugated to these types of antibodies. The second approach, back to what I mentioned previously, a genetically encoded approach where we can use fluorescent proteins targeted to cell structures and organelles. I'll speak to you in a few moments about a particularly useful gene delivery system called BACMAM. And again, a reminder that while we express these constructs in living cells, many of the fluorescent proteins are formaldehyde fixable if that's what's needed for your experimental design. Lastly, for lack of better phrases, this category I'll term fluorescent conjugates. This not only includes a variety of different classic, say, tracker dyes, for example, where we can exploit the chemistry that we've built into these molecules to have them selectively accumulate in different compartments, say, for example, the mitotrackers, which are cationic and lipophilic, and therefore accumulate in actively respiring polarized mitochondria, or lysotrackers, which are acidotropic probes, which will accumulate in acidic vesicles. But it's also truly conjugates as well. So we can think about fluorescent conjugates of toxins or receptor ligands that, by virtue of the specificity of these toxins or receptor ligands, we can use them as conjugates of fluorophores to visualize the structures to which they bind. Also, drug conjugates are an interesting use in this category. One needs to be mindful, of course, in using conjugates of drugs because of the obvious activity that these drugs will have on living cells. So typically, we recommend this approach for fixed endpoint visualization of cell structures in living cells. A great example might be something like tubulin tracker green, a paclitaxel or Taxol conjugate of a green fluorophore that allows us to visualize live cell tubulin, but because of the action of tubulin binding of this drug, it comes with a note of caution. Now, sometimes we're surprised, and I use the example of tubulin tracker green because more recently we've developed a longer wavelength version of this probe that we've termed tubulin tracker deep red. So we developed it so that it had longer wavelength fluorescence spectrum to free up shorter wavelength channels for other probes. We were pleased to have developed a probe that can label quite selectively live cell tubulin, typically about a 30-minute incubation. It's a bright and photostable fluorophore. Where we were surprised, though, is to see how well live cells tolerated this conjugate. Unlike tubulin tracker green, which is a paclitaxel conjugate, this conjugate of a deep red fluorophore is of a drug, a derivative called docetaxel. So it might be to do with the difference in the drug affinity, maybe a little bit less affinity in this conjugate, but in the end of it, the cells seem to tolerate this better than they do with tubulin tracker green. It enables longer-term viewing of tubulin networks, such as mitotic spindle formation you can see in the lower left. Even in neuronal samples, we're seeing very good tolerance of this probe in more sensitive cell types like neurons. I'll play another movie here. This is just HeLa cells, but you can see them dividing quite nicely after labeled with tubulin tracker deep red and imaged about every 20 minutes. But we're also seeing, these are live hippocampal neurons from mice that we can see look to be tolerating this probe quite nicely over about 72 hours, imaged at every 5 minutes. Okay, so with that, I'll go back to the fluorescent protein-based approach that I mentioned and share a little bit more information about the BACMAM gene delivery system that I mentioned before. If you're not already familiar with BACMAM, it stands for baculovirus with a mammalian promoter. So this is based on enhanced CMV, so we get very good expression in a wide variety of mammalian cell types. The exception here would be hematopoietic cells. We definitely have challenges expressing at any good efficiency in hematopoietic cells, so lymphocytes, macrophages, and such. However, in other mammalian cells, it's very easy to use. We really designed this to be more like a reagent in the typical workflow you might associate with using fluorescent proteins and doing upstream molecular biology. We can add more or less of these baculoviral constructs to cells to be transduced and control the transient levels of expression in these cells. It's non-toxic to us and generally speaking to cells that we transduce. It's non-replicating in mammalian cells. So really only BSL level 1 cell culture safety requirements are needed. It is flexible in that you can transduce large batches of cells and then cryopreserve them in aliquots for use later. And I think perhaps the most redeeming attribute of this gene delivery system is its amenability to transduce traditionally difficult to transfect cell types. A number of different primary and stem cell models we've had and others have had great success. And in particular in neurons, right? Very difficult to work with in terms of fluorescent protein transfections, in terms of toxicity and low efficiency of expression. We've found in a variety of different neuronal models, we get very good expression levels, anywhere from 50, 60% on up to better than 90% in some of these difficult to work with models. I'll show you an example of what this starts to look like in when transducing cells. These are two examples here of cells co-transduced with what we term cell light tubulin GFP. The cell light reagents are the fluorescent proteins that are for structural studies with those localization sequences for various structures. So cell light tubulin GFP along with cell light actin RFP. And when I play this movie, hopefully on your screen, you're able to see some of the dynamics that are available to us when we add temporal resolution into the mix when imaging cells. A lot going on here, right? When we look for longer periods of time using these same constructs, we're able to see cells dividing, mitotic spindle formation with the GFP tubulin as they divide over time. So really useful gene delivery system to be aware of. I will, in addition to cell light reagents, share with you technologies we've developed based on baculovirus, also termed primo sensors. These are just those functionalized fluorescent proteins for a variety of different readouts inside cells using this system. So I mentioned difficult to work with cell types. Here's just a couple of examples. In this case, iPSC-derived mouse cardiomyocytes expressing cell light tubulin or cell light actin. And I hope you can see on your screens this contractile activity that we can observe on the stage of a microscope. As expected in these cardiomyocytes, this contractile activity is expected. And then we can start to envision experiments where we can combine organic dyes. In this case, perhaps calcium indicators would be appropriate for this concomitant calcium bursts that are associated with the contractile activity in cardiomyocytes. I mentioned neurons, so I'll show you an example here from adult mouse adult mouse hippocampal neurons. Here just expressing an untargeted, what we call GFP transduction control. So this just fills these neurons with green fluorescence. Other neurons in this culture were transduced instead with cell light mitochondrial targeted RFP. So when I play the video, you not only will be able to see some cells and in particular axons filled with GFP, but also if you look carefully enough, you'll see mitochondria traveling along the axons of other neurons. So really interesting looks at cells that you can get using this gene delivery system that can be very difficult to do with traditional transfection techniques and also organic dye-based approaches. We can take closer looks and then start to get really nice views of growth cone dynamics, for example, in neuronal models. So real enabling technology that I think is good to be aware of. So let's go back to our cell health continuum and go on to another topic. How about cell cycle and cell proliferation? So here, again, we can use a fluorescent protein-based approach. I'll highlight a really clever approach that was originally developed by the great Atsushi Miyawaki. He and his lab at the Riken Institute in Japan developed a number of constructs that they termed the Fuchs cell cycle sensor. FUCH is actually an acronym for fluorescence ubiquitinylation cell cycle indicator. It's multiple constructs. Here I've highlighted geminin, a protein that is expressed during S, G2, and M. And then it is ubiquitinylated and tagged for degradation as cells exit mitosis. The second construct here is a protein called CDT1, here fused to RFP. It is expressed during G1 of the cell cycle, and like geminin, is tagged for degradation by ubiquitin, and then this protein is degraded as cells exit the interface between G1 and S. So the idea here is that we express these constructs in populations of cells, and as they divide and proliferate, we can get an approximation of where in the cell cycle they might be by virtue of the color of their nuclei. So, right, a really clever construct, a really interesting way to visualize temporally and spatially as cells proceed through the cell cycle. So we built this into baculovirus. So we deemed it here, of course, the primo FUCH cell cycle sensor because it's in a baculovirus construct. The image here you see is a fixed endpoint image reminding us that these fluorescent proteins can be formaldehyde fixable. So we see one cell expressing just green geminin, so S, G2, or M. Another cell red only, so G1 with CDT1. And then another cell that appears to have a yellow nuclei. This is just the overlay of green and red and indicates for us that this cell is in the interface between G1 and S. The purple signal here is just a wheat germ agglutinin conjugate to label the surface of cells. So we have a little bit of context. When we play a movie here in a U2S human osteosarcoma cell expressing FUCH by baculovirus gene delivery, we can see the parent cell divide, the daughter nuclei go colorless, and then as CDT1 comes up with RFP, we get a red color, and then it'll go yellow as geminin starts to come up in this interface between G1 and S. So again, a really clever way to visualize the cell cycle, and now we've made it more easy to use by introducing these constructs into baculovirus for the BACMAM gene delivery system. Now, for any level of scale, it can be far easier to use organic dye-based approaches, especially in high content imaging and analysis if we're interested in proliferation of cells. So I'll introduce to you a really useful chemistry platform that's termed click chemistry as a preface to some of these assays that we've developed for proliferation and other aspects of cell biology. So click chemistry was originally described almost 20 years ago now by Barry Sharpless. He used this terminology to describe a number of different reactions for specifically and efficiently joining together small subunits. And the term click chemistry stuck to one of these reactions, in particular the copper catalyzed azide alkyne cycloaddition. So what that means for us in cell biology is that we can introduce in live cells an azide containing molecule, and then subsequently after fixation and permeabilization, we can react that specifically with a fluorescent alkyne in the presence of copper. This will form a stable triazole reporter, and that's it, really. Now we can do the opposite. Instead, we can incorporate an alkyne into living cells and then subsequently detect with a fluorescent azide. Either way, it's click chemistry, and either way, it's important to appreciate, especially for proliferation assays, the small size of these labeling and detection partners. So if you look on the scale of size down below, we can see compared to large, perhaps antibody-based methods, the molecular modifications of alkynes and azides are quite small, and this can be a real important difference when access is at issue. So I'll contrast it to the traditional assay for proliferation based on bromodioxyuridine. This thymidine analog is given to cells in culture, and to the extent these cells are proliferating during that exposure, to the extent that they pass through S phase during that exposure, they incorporate bromodioxyuridine into nascent DNA. So that's great. We go in with an antibody against BRDU that's labeled with fluorophore to detect the incorporated BRDU. But again, the problem is access. So what we have to do is denature our samples, typically with hydrochloric acid, so that we can gain access of the incorporated BRDU with our bulky antibody reporter. And that's the assay. So it's non-radioactive, of course, unlike the very traditional approach of tritiated thymidine. It's multiplex compatible to some extent, right? But we're treating our samples pretty harshly to denature, so we can easily destroy epitopes that we may otherwise want to detect with other antibodies. Morphology can be compromised, and of course, if we're interested in DNA content measurements, those probes required for DNA profiling require double-stranded DNA, so that's off the table. So the click chemistry-based assay is very similar with a couple of very important differences. This is based on ethynyl deoxyuridine instead of BRDU. We have an alkyne group here instead of a bromine. And like BRDU, EDU is given to cells in culture. If they pass through S phase during that exposure, they'll incorporate EDU and this alkyne handle into nascent DNA. And then without any kind of denaturation step, we can, in the presence of copper, run a simple, typically about a 30-minute click reaction with fluorescent azides and click together these partners for a stable triazole reporter. And that's it. So there's no DNA denaturation required. The protocol is drastically simplified. And this is compatible with other probes such as antibodies or dyes that require double-stranded DNA. Our newest version of click chemistry called ClickIt Plus is also compatible with fluorescent proteins that are otherwise susceptible to quenching in the presence of copper. Copper is a catalyst required for this reaction. So we've leveraged copper chelation technology to make newer versions of click chemistry, that is ClickIt Plus technology, compatible with fluorescent proteins. In actual imagery, it looks something like this, where this is an example of muntjac skin fibroblasts that were exposed to EDU. We can see here two pinkish labeled nuclei that are EDU positive, so proliferative cells in the presence of cells that did not incorporate EDU during that exposure. And importantly here, these fragile structures like the tubulin network, the Golgi apparatus, and peroxisomes labeled with a variety of other probes are left intact during this gentle protocol involving click chemistry. It can be used in whole animals, such as this example showing a section from rat ileum. So this intestinal section was, well, first of all, animals were fed EDU, and then this intestinal section was taken, and then click reaction was performed, and we can see these red proliferative nuclei that are EDU positive amongst the total nuclei that are just labeled with DAPI. A fun example I like to show here is from one of the pioneers of click chemistry, actually, Adrian Salik's group here in the US. They published this years ago now in PNAS, showing the detection of proliferative cells in sections of rat brain using click chemistry. So really a very robust assay that has been well validated throughout the literature and by us in a number of different systems, mammalian and otherwise, across kingdoms. This technology, like many of the technologies I'll show you, are perfectly amenable at scale using high content screening. This is an example just showing a simple dose response curve of the percent positive EDU cells against dose response of a toposide, a known inhibitor of cell proliferation. And using our Cell Insight high content screening platform, we can show not only quantitative data analysis here in the dose response curve, but we can also associate any of those data points to actual images. And we can see here an increasing concentration of the toposide shows in both counts an inhibition of cell proliferation by a decreased incorporation of EDU. So I talked about proliferation using click chemistry, but again, a number of different other assays for cell biology can be done using click chemistry. We can do a similar assay using ethynyl uridine instead of ethynyl deoxyuridine. EU will allow us to visualize global transcriptional levels in populations of cells. We do the same thing with modified and clickable amino acids such as homopropargylglycine or azeto-homoalanine, using these modified amino acids to look at nascent protein synthesis. Post-translational modifications can be interrogated using click chemistry, particular metabolic sugars that are azide modified. We can even look at damage in cells, DNA damage, such as the tunnel assay for DNA nicks associated with apoptosis can be visualized using click chemistry, and even aspects of cell biology and stress like lipid peroxidation using clickable forms of polyunsaturated fatty acids. Okay, so let's return then to our cell health continuum and talk a little bit about another very important pre-lethal mechanism in cell health and stress, that is oxidative stress. Of course, we know that the generation of reactive oxygen species is normal in aerobic metabolism, and not only a byproduct of aerobic metabolism, but also very important signaling aspects in cell biology. We do know that once our cell's natural antioxidant mechanisms like reduced glutathione, for example, SODs are overwhelmed, that is when oxidative stress really emerges and, of course, has been implicated in a number of different important areas like cancer, cardiovascular disease, diabetes, neurodegeneration. The list goes on and on, even important in aging, right? We also know that oxidative stress is a very good predictor of cell toxicity in screening for potential therapeutics, very predictive in cell-based measurements of what might happen in human biology. We also know something about where ROS come from. Of course, NADPH oxidases, the so-called NOXes, lipooxygenases, and of course the central hub for mitochondria or for ROS in the mitochondrial electron transport chain. These respiratory chain complexes, of course, reside in these inner mitochondrial membrane folds called the cristae. So here in the bottom of the slide here, the very bottom where the Krebs cycle would occur, reduced pyridine nucleotides are produced by the TCA cycle, like NADH. NADH is an energetic substrate at complex 1 here of the electron transport chain. So as NADH is oxidized, electrons are passed down to subsequent respiratory chain complexes and mobile carriers like ubiquinone and cytochrome C, ultimately reducing oxygen to form water. But as these reactions happen through complexes 1, 3, and 4, protons are translocated from the matrix to the intermembrane space, creating an electrochemical gradient across the intermitochondrial membrane that, of course, drives the phosphorylation of ADP to make ATP at complex 5, but also this electrochemical gradient across the intermitochondrial membrane can be exploited with use of probes like mitotrackers or TMRM and the like, so that we can get a relative indication of mitochondrial membrane potential and overall mitochondrial and cell health by using these probes. So really important area here in cell health. And again, oxidative stress here is a big factor. So this electron transport system, oxidative phosphorylation, is very efficient but not perfect. And when oxygen is incompletely reduced, we form superoxide, which can quickly form peroxynitrite in the presence of nitric oxide. But even if mitochondrial SODs dismutate to peroxide, Fenton chemistry can catalyze the formation of hydroxyl radicals, highly reactive and damaging a reactive oxygen species, right? So there's a lot going on here in terms of cell health and cell stress and cell death. Traditionally, we've probed this phenomenon of oxidative stress in living cells using reduced fluoresceins, but this approach has some severe limitations, not the least of which is the photostability of fluorescein once oxidized from reduced fluorescein. Also, we've observed that photooxidation can occur with these probes, which is an obvious problem for a falsely positive signal. And also, in order for these probes to permeate living cells, they have to be acetylated. And because esterase activity can be present in serum of complete media, it's recommended that we remove cells from complete media just to load reduced fluoresceins. So this can be stressful on cells as well. So we sought out some years ago now to develop improved probes for oxidative stress. And the first one that we developed is called CellRox Deep Red. So one immediate advantage here, if you look at this example in the upper left of endothelial cells stressed with menadione, is that we've loaded these cells with CellRox Deep Red in complete media. So these are passively permeable to living cells. They're not acetylated, so they can be loaded in complete media. You can see with a menadione challenge the robust response we can achieve in endothelial cells, and we can prevent that response using superoxide scavengers, for example, here manganese TBAP. Also notice the improvement in photostability. So compared to this green photobleach curve using reduced fluoresceins, the CellRox Deep Red holds very stable across continuous exposure in microscopy. We were surprised, delightfully so, to see that we can retain CellRox Deep Red signal, not permanently, but for at least a couple of hours You can see on the upper panels on the right here, the live response again we get with the metadione challenge, but we can preserve that response quite nicely after formaldehyde fixation. Again, only for a couple hours, but this can be the difference getting across a 96-well plate or even several plates, which is a real improvement compared to traditional probes for reactive oxygen species. So we went on to develop other CellRox probes, namely CellRox Orange and CellRox Green. CellRox Green was a little bit different because it's actually a reduced nucleic acid stain, so not only requires oxidation, but also binding to nucleic acid to fluoresce appreciably. I like CellRox Green a lot because it will, unlike CellRox Orange, will survive fixation, and then beyond CellRox Deep Red will actually survive detergent as well. So this can be multiplexed with probes like antibodies that require detergent fixation and detergent permeabilization. So here's an example of human osteosarcoma cells that were loaded with not only CellRox Green, but also a mitochondrial membrane potential probe called TMRM, or tetramethylrhodamine methyl ester, that fluoresces after accumulating in actively respiring mitochondria here in the red. We'll see after a metadione challenge here that the mitochondria depolarize, we lose the red fluorescence, and then start to gain green fluorescence in the nuclei of these cells as CellRox Green is oxidized and bind nucleic acid. So a real positive step for improvements in interrogating oxidative stress in living cells. Good. So let us return to our cell health continuum and cover a couple of more topics. The next being apoptosis, of course, a very important area for many aspects of cell biology. In terms of parameters to probe whether cells are signaling apoptosis extrinsically through fast ligand or intrinsically through the mitochondria. A reliable marker of caspase-dependent apoptotic cell death is activation of these downstream caspases 3 and 7. So we designed a probe that's based on an established peptide substrate for these caspases, DVD, and we've attached this peptide substrate to a DNA binding dye. So really it's a matter of adding this probe that we call cell event caspase 3,7 green to cells in culture, typically about a 30-minute incubation. And then once caspases 3,7 are activated, they'll cleave the DVD substrate and release the DNA binding dye to bind nucleic acid and fluoresce green. So we can measure the green fluorescence beyond background and get a very good indication qualitatively and quantitatively on our percent apoptosis in a population. Qualitatively, it looks something like this. Again, we've combined this with TMRM for mitochondrial membrane potential. Here in HeLa cells challenged with staurosporin, we can watch the loss of mitochondrial membrane potential as cells undergo stress and apoptosis. And then as caspases 3 and 3,7 are activated, we see an emergence of the cell event caspase 3,7 green signal. As I mentioned, we can use this and other probes quantitatively, again, using our Cell Insight CX7, in this case, laser-based system. We interrogated a number of different compounds, a subset of the so-called killer plate. I'm showing down on the left here an example positive control staurosporin dose response curve plotted against the percent positive cell event cells. The heat map up top really just shows where if we achieved or if we observed a 3-fold or better increase in the percent positive in the population, we gave it a red box, and if we did not, then we gave it a green box. So a gray box. So the idea here is that we're able to really scale up and quantitatively interrogate whether or not cells might be pro or anti-apoptotic in behavior. So far, I've shared with you a number of different probes in 2-dimensional models, 2D monolayers of cells. I'd like to now switch gears a little bit and show some of these probes in action in 3D models. So I'll highlight in particular 3D models that are tumor spheroids. And of course, it's been well established that these 3D spheroid cultures are fundamentally dissimilar than monolayer cultures. It's kind of pointing out the obvious, but really it's important to mimic, in this case, solid tumors as best we can for physiological relevance. There's a number of reasons that we can explain these differences. and not only explain differences in terms of biology, but it's been reasonably well established in the literature that these 3D cultures better mimic solid tumors in terms of pharmacology. Even some publications that claimed that these 3D models display better predictive value. But really, the basis of the differences comes down to really gradients in metabolites, gradients in oxygen. It's generally accepted that the periphery of these 3D spheroids are more proliferative, whereas the core of these spheroids are more dead cells, be it necrotic or apoptotic. I'm showing just a couple of qualitative examples here using our Cell Insight high content screening platform. On the left, we've exposed these spheroids to hypoxia and labeled them not only with hooks for all the nuclei, but also what we call the Imagit green hypoxia reagent. This is essentially a nitroreductase substrate bound to a fluorogenic green dye. And so it's reasonably well accepted that when cells undergo a shift to hypoxic metabolism, nitroreductases are upregulated. And so we're seeing nitroreductase activity reflected in this green signal. On the right, using the cell event caspase 3,7 green probe that I just described, we're showing a predominantly apoptotic population of cells in the core of this spheroid, otherwise labeled with hooks for total nuclei. So we can start to combine these technologies, that is high content imaging and some of these fluorescent probes. Here is an example from our Cell Insight system again, showing not only a fluorescence interrogation in the spheroid of apoptosis, showing a dose response after treatment with niclosamide, a dose response of niclosamide showing an increase in the cell event caspase 3,7 green signal that's dose dependent, but also a dose-dependent decrease in the size of the tumor spheroid that we can measure in bright field using the Cell Insight system. So we can look quantitatively at the top and the same effect really qualitatively in these bottom panels with increasing levels of apoptosis as we increase the dosage of niclosamide. We can start adding parameters here. I'm showing two different spheroid models, A549 and HeLa cells cultured as tumor spheroids, and an increasing dosage of niclosamide. So up top with no drug or very little drug, we see mitotracker orange being used here to show plenty of mitochondrial membrane potential, in particular in the periphery of these tumor spheroids. And as we increase the dosage of niclosamide, we're seeing a loss of mitochondrial membrane potential, but an increase as expected in apoptotic cell death. And again, we can represent this data quantitatively as well with respect to both parameters and with respect to both models. Here we can see that on the dose response curves on the bottom of the screen. Okay, so I'll share a few other applications with you, but before I dive into some of the immuno-oncology applications, I want to remind people of a very simple approach that I mentioned earlier in the webinar, the use of cell trackers to label whole cells for a variety of applications. So again, before we go on with some immuno-oncology applications, some other applications and just the reminder of the use of these live cell permeable either amine or thiol-reactive fluorescent dyes, the so-called cell trackers or cell trace probes. These really are just live cell permeant protein stains, again, that can be used in a variety of applications. On the left here, I'm showing samples of endothelial cells that were seeded on our Geltrex matrix so that they are able to differentiate into pseudovasculature as you would do for an angiogenesis assay. As I play this movie that was generated on our EVOS M7000 imaging system, what we'll see is a differentiation of these cells from an endothelial cobblestone-like phenotype to these pseudotype or pseudovasculature tube-like structures. We're using cell tracker deep red in this case, but we've also labeled the sample with cell event caspase 3,7 green. So if you look carefully, you'll see some green dots here indicating the nuclei of cells that have been excluded from this differentiation process and have undergone apoptosis. Another common example for the use of cell tracker dyes is for wound healing. Here on the right, we've used cell tracker red, labeling fibroblasts in a plate where we've induced a scratch wound, and then we can, we can visualize these cells healing, so to speak, this wound that we've induced. Again, just using cell tracker red probe for a very simple labeling experiment. Again, collected on our M7000 imaging system that really makes it easy to generate these kind of movies. We can get quantitative on our EVO systems as well using our Celeste software. In the case of wound healing, we can easily just locate wound boundaries and then do a calculation over time for when we might achieve maybe a 50% closure. So a lot you can do here quantitatively, not just qualitatively. So using cell trackers, again, can be applied to applications for immuno-oncology. So I'll share a couple of examples here with you. This is the case of where we have cultured A549 lung cancer spheroids in nunclon low attachment plates. Very important because we want these cells to stick together, not to the plastic substrate. So these nunclon spheroplates are really not only ideal, but really necessary for growing high quality spheroids in culture. So what we've done here is we have exposed these tumor spheroids to either activated T cells, activated with CD3, CD28 beads, or not, just peripheral T cells that have not been activated. These T cells were labeled with cell tracker deep red and applied to these tumor spheroids. We've included cell event caspase 37 to the mix here, and you can see in the absence of T cells, we do have some apoptotic cell death in the spheroid. When we apply activated T cells versus inactive T cells, you can see a pretty dramatic difference in the level of cell killing as indicated by cell event here. We can get quantitative here rather than a dose response of a drug. We're showing a dose response really of T cells, so a difference in the target to effector cell ratio. And of course, as we increase the number of T cells, we're going to get an increase in the cell tracker deep red signal simply because there's more T cells. But importantly here, we're seeing an increase in apoptotic cell death as we increase the effector to target cell ratio in this experiment. Again, using our Cell Insight CX7 high content screening platform. Okay, so another example here, but in this case using NK cells, we're showing the application of NK cells isolated from peripheral blood to SKBR3 spheroids, here labeled with Nuke Blue Live, which is just a room temperature stable, convenient formulation of Hoechst. For total nuclei here, in the absence of NK cells, we're seeing very little, if any, apoptotic cell death. But when we apply cell tracker deep red labeled NK cells, we see an increase in apoptotic cell death. When we include trastuzumab, an antibody that will help facilitate this killing process, we see, as would be expected, a dramatic increase in apoptotic cell death, but also really a compromise in the structural integrity of this tumor spheroid. All right, so to close out here, I'll share with you one last topic from the cell health continuum, not just phagocytosis, but really the general idea of internalization in cell biology. And whether we're dealing with phagocytosis or endocytosis, ligand internalization or antibody internalization, all of these internalization pathways share in common an acidification of cargo as it's internalized. So we can exploit that acidification process by using fluorogenic pH-sensitive probes. And what we've developed here that are real improvement over traditional approaches are termed Frodo dyes. So this is short for pH-sensitive rhodamine derivatives. And whether we're talking about red or green Frodo dyes, you can see here a pH-dependent increase in the fluorescence of the Frodo red or Frodo green. That is, as the acidity increases, so does the signal of these probes. What's also very nice here is that the pKa of these sensors is right around neutral, about 6.8. So that's important because that's about the pH threshold going from extracellular to intracellular environment. So this really results in a no-wash assay for a variety of different internalization assays. One context in which we can use these is with phagocytosis of bacteria. Here I'm showing as bacteria labeled with Frodo dyes are internalized and are exposed to the relatively acidic environment of the phagosome, they fluoresce. So I'll show you a couple of movies here. This is heat-killed staph conjugated to Frodo red, fed to macrophage-like cells. And we can see only after internalization of this cargo do we see an emergence of the Frodo red signal. Similarly, when we conjugate zymosan yeast particles to photo-green, we see an emergence of green photo signal only after internalization of these particles. Now, it's important also to be able to label antibodies with functional fluorophores like this, but it's also important to make sure that we maintain not only affinity of the antibody for its target after conjugation, but in this case, the functionality of the pH sensor after conjugation. So we can see here, trastuzumab labeled with photo-red or photo-green, the newer IFL derivatives of photo dyes, we can see a maintenance of a nice pH response here. And that wasn't a given. The classic photo dyes, we observed a severely blunted pH response after conjugation, and that's actually one of the observations that inspired us to develop these next generation IFL photo-red and photo-green probes for antibody conjugation. So once we've established this, we can go to work with these conjugates. Here I'm showing an endpoint image of trastuzumab conjugated to IFL photo-red. And we've concluded that this cargo has accumulated in lysosomes because we co-localized that red cargo with lysosomes that are visualized using the cell-light lysosomal targeted GFP. Some counterstains here in nuclear live for the nuclei, as well as the cell mass deep red stain for just a contextual label for the plasma membrane of these cells. Now, in terms of labeling antibodies, it's also very important in some cases to label the FC region of our antibodies so that we don't disrupt the specificity of the antibody for its target. So this is a really useful conjugation technology that we actually call site-click technology. It's site-specific antibody labeling based on a modified click chemistry. So the workflow is as follows. We start with our IgG of interest, and then we'll modify that antibody by first digesting terminal galactose residues using beta-gal, and we replace those residues with azide derivatives of sugars here using gal-T. So after this modification step, we're left with an azide-activated IgG, and that activation, that modification is on the FC tail, importantly. Then, using this azide handle, we can react to it probes like a modified IFL photo-red using special click chemistry that doesn't require copper catalysis that can disrupt our antibody. S-dibo just refers to strained ring chemistry and a soluble linker that we use in site-click chemistry to covalently label IgGs of interest and specifically on FC tails. So when we do that, we can do interesting experiments like incubation of these therapeutic antibody conjugates in tumor spheroids. So here we've used, in a so-called bystander assay, a mix of HER2 positive and HER2 negative cells in the spheroid. And as expected, we see internalization of the IFL photo-red trastuzumab in some cells, presumably the HER2 positive cells, but not in others, presumably the HER2 negative cells. When we go to a pure cell culture, that is of all HER2 positive cells, and do this experiment, we see a drastic uptake is indicated by photo-red, IFL photo-red. But we've also incubated here with Celavent caspase 37 green, and we see very little apoptotic cell death as expected because we're only just delivering the antibody, not a toxic payload like we would in an antibody drug conjugate. We can use site-click chemistry to do just that. That is, conjugate an IgG with not only a fluorophore reporter, but also a toxic payload. And when we do that in these tumor spheroids, we not only see that uptake as indicated by IFL photo-red, but also apoptotic cell death as we deliver that toxic payload using Celavent caspase 37. So really useful technologies to be aware of. That is the Frodo pH sensor platform, but also site-click chemistry for site-specific conjugation of antibodies. So with that, I'll leave you with some valuable resources that you can find online. The Bioprobes magazine is a great reference that we've been publishing for years now, usually about once or twice a year, that highlights what we're up to in R&D and then some nice highlights from the literature. If you're interested in reviewing some basics about imaging, flow cytometry, or just fluorescence in general, we've developed an educational website called the Molecular Probe School of Fluorescence. If you're interested in learning more about culture and model development of 3D models, we've developed a free e-learning course with that focus you can check out here. And then I always like to recommend our fluorescence spectroviewer, a really invaluable tool for ensuring that there is not only spectral compatibility of fluorophores you're considering in a multiplex experiment, but also compatibility with the instrumentation that you have on hand. So check that out as well. And thank you very much for everyone's attention. I'll leave my email address on the screen. I'm happy to take questions now, of course, but if questions come up in the future, feel free to email me. So thank you once again.
Hi, Mike. This is Barb, the host. Thank you very much for an outstanding presentation you gave. There are no questions on the Q&A board, but I am going to allow all of our guests to talk. All they need to do is unmute themselves. And if there's any questions, please just unmute yourself and you can direct them now to Mike. Well, I guess you did a really outstanding job, Mike. You don't even have any questions. We did have a few other attendees, but some seem to have dropped off at this point. But I know that my director just said, what a fantastic presentation. So thank you very much. I will post this on the GRCF website for anyone to view if they want to see more detail and review what was presented today, and also Mike's email address. Also, please keep in mind that the products referenced throughout this presentation are available at the core store at the Genetic Resources Core facility. Again, thanks everyone for joining. We very much appreciate your attendance, and please keep your eye out for future presentations at the Genetic Resources Core facilities. Thanks.
Okay, Barbara, actually, I think we have a question on the chat.
Oh, okay, I do.
Yeah.
You want to ask
George?
Yeah, so great question, really, especially in these strange times. So we don't have COVID or even viral particles that are already conjugated to the photosensors, but we do have reactive forms of the photo dyes, thiol and amine reactive forms of these dyes that allow for you to conjugate your own viral particles. And there is this has been published in general for conjugation of viral particles. And if you'd like more information on some of the protocol details that we've learned here at ThermoFisher Scientific, please email me and I'll provide that. But no pre-conjugated viral particles off the shelf, only the reactive forms of the photo dyes for you to make your own. Great question.
Are there any other questions? Okay, I think that about wraps it up. Thanks you very much, and we'll be in touch soon. Thank you, ThermoFisher team.
Thank you.